Welcome class. In this session, we'll be discussing regarding light reaction of photosynthesis. So that is under the mechanism of photosynthesis. So under the mechanism of photosynthesis, there are two steps. One is light reaction and another one is dark reaction. So here we'll discuss regarding light reaction, which is also called as Hill's reaction. So that is the another name for the light reaction of photosynthesis, because it has been first explained by the Robert Hill. So it is also called as Hill's reaction. And this light reaction, what is meant by this light reaction of photosynthesis? So these are the reactions that are dependent on the light. That is, so these reactions, they require light energy. So such type of reactions which are dependent on the light, they are called as light reaction. And the site of reaction will be thylakoid membrane, so which is a component of a grana. So each granum will be composed of thylakoids, and each thylakoids will be having a double membrane structure within which the photosystem will be present. So hence thylakoid membrane, they act as a site of light reaction. So that is regarding the introduction to light reaction. And this light reaction, it involves 4 different steps. That is photoexcitation of chlorophyll, photolysis of water or photooxidation of water, then photophosphorylation and photoreduction. So what is meant by photoexcitation of chlorophyll molecule? So in case of photoexcitation, so here there will be the reaction center. So let us consider P680 as well as P700. So that is the reaction center of photosystem 2 as well as photosystem 1 respectively. So when the light energy falls on this photosystem, so H nu refers to incident radiant energy or the light energy. So when light falls on the photosystem, so this photosystem will be composed of chlorophyll molecules associated with the electrons. So when the light energy falls on this particular photosystem, the electrons that are present in this particular photosystem, so they get excited to the higher energy state. So this forms the high energy electrode, so that we can represent it as P680 in the aster. So that indicates that it contains the excited electron or energy of electron of high energy. Same happens in case of photosystem 1 also. So it becomes excited, or it contains the energy, it contains electron that has been excited to higher energy level. So this process is called as photoexcitation of chlorophyll molecule. And as a result of photoexcitation here, electron hole will be created in case of both the photosystem, because electron which are present in the ground state, they have excited to higher energy state, whereas in ground state there will be deficient of electrons. So that results in the formation of the electron hole. So that is regarding photoexcitation of chlorophyll molecule. And next step is regarding photolysis or photooxidation of water. So in this mechanism, so what happens? The water molecule, in the presence of light energy, as well as an enzyme called complex called as oxygen evolving complex, which will be having manganese as a central metal ion as well as the chloride ion. So in the presence of oxygen evolving complex, the water molecule will be split into hydrogen, 2 hydrogen ions, as well as a molecular oxygen, and 2 electrons will be released. So this is called as photooxidation or photolysis of water, where the water molecule will be split into hydrogen as well as the molecular oxygen along with the electrons. So this will be catalyzed by the enzyme called as oxygen evolving complex, which will be having manganese as well as the chloride ion as a enzyme activator, which catalyzes this reaction. So manganese activates the enzyme complex, whereas chloride, it carries this electron in order to fulfill the electron hole over here. So that is regarding the photolysis or photooxidation of water. And next one is regarding photophosphorylation. So generally the term phosphorylation refers to addition of phosphate to any compound, that is called as phosphorylation. So generally when we speak about phosphorylation, we speak in terms of synthesis of ATP from the ADP. When ADP is added with the phosphate, so it will form the ATP, and this process is generally termed as phosphorylation. So here the term photophosphorylation refers to addition of phosphate to adenosine diphosphate to form ATP, that is adenosine triphosphate. It occurs in the presence of sunlight or the light energy. So that's why it is called as photophosphorylation. And in case of this photophosphorylation, there are mainly of two types. One is cyclic and another one is non-cyclic photophosphorylation. So regarding that, we'll discuss in detail for after discussing these steps and all. And next is regarding photoreduction. So photoreduction is a phenomenon where the reducing agent will be formed. Okay, it will be reduced. So here the reducing agent used will be NADP. So where NADP refers to nicotinamide adenine dinucleotide phosphate. So full form of NADP is nicotinamide adenine dinucleotide phosphate. So that is the full form of NADP. So this NADP will be used as a high energy compound or a reducing agent, which in the presence of two hydrogen ions, it will be reduced, and two electrons. So it will be reduced into NADPH2. Okay, so this process, it also occurs in the presence of light energy. So hence it is called as photoreduction, whereas high energy compound will be reduced in the presence of sunlight, and that high energy compound will be NADP, which will be reduced into NADPH2. So these are the, and this particular reaction, photoreduction, it will be catalyzed by the enzyme that is called as NADP reductase. So this NADP reductase, it catalyzes the conversion of NADP into NADPH2, and these are the main steps that are involved in the light reaction of photosynthesis. So these steps can be studied in detail, or it can be represented schematically by referring to non-cyclic as well as cyclic photophosphorylation. So as I said, in phosphorylation, there are two types. One is cyclic and another one is non-cyclic. So first we'll look into the non-cyclic photophosphorylation. So in case of non-cyclic photophosphorylation, and as it is mentioned in case of photosystem, when we studied about difference in photosystem, we have said that non-cyclic photophosphorylation, it involves the participation of both photosystem 2 as well as photosystem 1. And it is called as non-cyclic photophosphorylation. That is because electron lost here during the photoexcitation, it won't come back to the same reaction center. Electron lost will not come back to same reaction center. So that's why it is called as non-cyclic photophosphorylation. Electron lost will be not cycled back here. So what happens in non-cyclic photophosphorylation? We'll see. So in case of non-cyclic photophosphorylation, so when light energy falls on the photosystem 1 and photosystem 2 simultaneously, so as usual, excitation of the chlorophyll takes place. So photoexcitation takes place where the electron, it will be moving from ground state to higher energy state, resulting in the formation of the electron hole in case of both the photosystems. Okay, so now the high energy electron that is formed in the photosystem 1, it cannot be retained in a higher energy state for the longer duration. Either it has to come back to the ground state or it should be accepted by the electron carrier. So here the excited electron, it won't come back to its original reaction center. So that's why upon excitation of the electron, so this electron will be accepted by the primary electron acceptor. And this primary electron acceptor involved in non-cyclic photophosphorylation is called as pheophytin. So pheophytin, it accepts the high energy electron and it gets reduced. And upon reduction, again pheophytin should be ready to accept the another electron that is coming during the photophosphorylation or during the light reaction. So hence the electron acceptors that are involved in this reaction, they undergo series of oxidation reduction reaction, forming the electron transport system here. Okay, so once the electron is accepted by the pheophytin, now this now this electron will be transferred to the one more electron acceptor that is called as plastoquinone. So in this particular electron transport system, there are two types of electron carriers. One are fixed or the immobile electron carriers, and another one are the mobile electron carriers. So this plastoquinone, plastocyanin, all this act as a mobile electron carrier which can move from one electron carrier to another one. Whereas the pheophytin or cytochrome B6F complex, all these forms the fixed electron carrier where they are fixed in their position. So once the electron is received by the pheophytin, once the electron from pheophytin is received by the plastoquinone. So this plastoquinone, in order to uptake electron, it also uses the hydrogen ion from the stroma of the chloroplast for the complete reduction. So hydrogen ion will be richly present in the stroma of the chloroplast. So once this plastoquinone accepts the electron, along with the electron, it also accepts the hydrogen ion. At a time, it accepts two hydrogen ion for the complete reduction of this plastoquinone. So there are two types in plastoquinone, type A as well as B, QA and QB. So these two will accept hydrogen ions along with the electron and they get reduced. And once they are reduced, now they will pass on their electron to the cytochrome B6F complex. And as here electron is transported to cytochrome B6F complex, the two hydrogen ions that are accepted by the plastoquinone for its reduction, it will be pumped to the lumen of thylakoid. So it was accepted from the stroma and it has been released into the lumen of thylakoid. So you know what is meant by lumen. That is the space that is present inner to the thylakoid membrane is called lumen of thylakoid. Okay. So this hydrogen ions accepted by the plastoquinone will be pumped to the lumen of the thylakoid. So now your cytochrome B6F complex has accepted the electron. So that electron will be now transferred to the one more electron carrier called as plastocyanin. And this plastocyanin is an electron carrier which can diffuse between photosystem 2 as well as the photosystem 1. Okay, so from cytochrome B6F complex, electron will be carried to the photosystem 1 through the mediation of plastocyanin, so that here initially the electron has been lost in case of photosystem 1 due to photoexcitation. So that will be fulfilled by the electron received by the plastocyanin. So now we'll see what happened to the excited electron in photosystem 1. So the high energy electron or the excited electron in photosystem 1, it will be accepted by the series of electron carriers such as chlorophyll A as well as A0 and A1. So which are considered as modified chlorophyll A molecules, and further electron will be accepted by the iron sulfur cluster or the FeS protein. And later this electron will be transferred to the ferredoxin. So it is one of the reducing agent. So it is one of the electron acceptor or the reducing agent. So once the electron is accepted by the ferredoxin, so this ferredoxin, it will be associated with the enzyme complex that is called as NADP reductase. So this ferredoxin will be associated with this NADP reductase, and once electron comes to ferredoxin, So electrons from the ferredoxin will be donated to the NADP reductase enzyme, so which catalyzes the conversion of NADP plus into NADPH2. And for this, 2 additional hydrogen ions will be accepted from the stroma. Okay, so that is how photoreduction takes place in case of non-cyclic photophosphorylation. So far we have seen how the excited electron from photosystem 2 has been finally used in the photoreduction of NADP to form NADPH2. But so far nowhere we have spoken about how the electron hole created in photosystem 2 is fulfilled here, right? So here, initially we have discussed, so photosystem 2 is associated with the oxygen evolving complex. And the role of this oxygen evolving complex is to split the water molecule into 2 hydrogen as well as the molecular oxygen along with 2 electrons. So upon splitting of water, whatever electron is formed here, so this electron will be pumped to the photosystem 2 and reaction will be mediated by the chloride ions. Okay, so that is how the lost electron in the photosystem 2 during the photo excitation will be fulfilled, or it will be replenished. So that is how electron hole created during non-cyclic photophosphorylation will be fulfilled in the photosystem 2. So that is regarding the non-cyclic photophosphorylation. And here this hydrogen ions that are formed, so it also will be released to the lumen of the thylakoid itself. So that is regarding the non-cyclic photophosphorylation. So next is regarding cyclic photophosphorylation. So understanding cyclic photophosphorylation is simpler. That is because it only involves the participation of photosystem 1. That is, the reaction center will be P700. So here what happens? So in case of photosystem 1, when the light energy falls on the photosystem 1 in cyclic photophosphorylation, so that electron electron present in the reaction center, it gets excited. Okay, excited electron will be accepted by the A0, A1 complex. These are the modified chlorophyll molecules. And further it will be transferred to the ferrous sulfur protein. And from there it will be transferred to the ferredoxin. And from the ferredoxin, so this electron will be carried by the plastoquinone. Okay, so we have seen in non-cyclic photophosphorylation, from ferredoxin electron will be transferred to NADP reductase. But here in cyclic phosphorylation, that process is not there, or that enzyme is not there. So electron from the ferredoxin will be transferred to the plastoquinone. So you know that plastoquinone, it accepts hydrogen ions along with the electron, and this electron will be further transferred to the cytochrome B6F complex. Okay, and from the cytochrome B6F complex, so electron will be accepted by this and hydrogen will be pumped to the lumen of thylakoid. Okay, and from this cytochrome B6F complex, electron will be accepted by the plastocyanin, and this plastocyanin now carries this electron back to that of the photosystem 1, or to the reaction center of photosystem 1. So here what happens, whatever electron is lost initially, it comes back to the same reaction center, or it will be cycled back to the same reaction center. So that is why it is called as cyclic phosphorylation, where the electron lost will be cycled back to same reaction center. And here this is not involved with the photolysis of water, is not seen here, photoreduction is not seen here, only the electron will be recycling here in case of cyclic photophosphorylation. So that is regarding the cyclic as well as the non-cyclic photophosphorylation that is involved in the light reaction. So why we are discussing the cyclic and non-cyclic photophosphorylation here in detail? That is because this photophosphorylation, it is coupled with the synthesis of ATP. So far we have just studied the cyclic and non-cyclic photophosphorylation. Nowhere we have discussed regarding ATP synthesis. So next, in the next part, we'll look into the synthesis of ATP from the as a result of cyclic and non-cyclic photophosphorylation. So in case of non-cyclic photophosphorylation, if we see, so for each molecule of water, so totally 4 hydrogen ions will be added to that of the thylakoid lumen, right? So that is the total number of hydrogen ions that are added to the lumen of thylakoid during non-cyclic photophosphorylation. 2 from the splitting of water, 2 hydrogen ions from the transfer of electron from plastoquinone to cytochrome B6 F complex. Totally 4 hydrogen ions will be added to lumen of thylakoid for splitting of 1 molecule of water. Whereas in case of cyclic photophosphorylation, if we see, for a molecule of water, only 2 hydrogen ions. So per molecule of water, 2 hydrogen ions will be added to that of the lumen of the thylakoid. So by keeping this in the mind, so now we have to see totally how many hydrogen ions will be added from the stroma of chloroplast to lumen of thylakoid during the light reaction. Okay, so we know the equation of photosynthesis. So it uses 6 molecules of carbon dioxide and 12 molecules of water to form a carbohydrate, as well as 6 molecules of oxygen and 6 molecules of water. So this is the equation of photosynthesis. So I said that in non-cyclic photophosphorylation, what happens? For 1 water molecule, 4 hydrogen ions will be added to the lumen of thylakoids. So for 12 molecules of water, how much hydrogen ions will be added? It will be 12 into 4. It will be 48 hydrogen ions, right? Whereas coming to cyclic photophosphorylation, so for a molecule of water, 2 hydrogen ions will be added to the lumen of the thylakoid, right? So here 12 molecules of water is used in the photosynthesis. So totally it will be, so how many hydrogen ions that are added here? So it will be 12 into 2. So that will be up to 24 hydrogen ions. And in addition to that, it also involved in the synthesis of 12 NADPH2 molecules also. So that is regarding the end products of the photophosphorylation. Okay. So if we see here totally from non-cyclic and cyclic photophosphorylation, so in the lumen of thylakoid, we will be having up to 78 molecules of hydrogen ion and lesser number of hydrogen ion in the stroma. So we know that every molecule in a cell or a system, they tend to move from the region of higher to lower concentration, right? So here the concentration of hydrogen ion will be more in the lumen of thylakoid compared to that of the number of hydrogen ions in the stroma. So as a result, what happens? This hydrogen ions, they tend to move back to the stroma of the chloroplast. What happens due to movement of hydrogen ions to the stroma of chloroplast? How it is coupled with the ATP synthesis? So it can be explained through the chemiosmosis hypothesis, which was proposed by the Peter Mitchell. So Peter Mitchell, he gave the concept of chemiosmosis hypothesis, which explains about the ATP synthesis in case of photosynthesis. Same concept is used in the respiration also. So according to Peter Mitchell, so here chemiosmosis, it refers to osmosis, you know, movement of solvent molecules from region of higher to lower concentration. Whereas chemiosmosis refers to movement of the protons or the hydrogen ions from the region of higher to lower concentration through the thylakoid membrane. So this thylakoid membrane is also selectively permeable. So that is called as chemiosmosis. So it is concerned with the movement of hydrogen ions. And according to this chemiosmotic hypothesis, it says that every 2 hydrogen molecules that are pumped from the lumen to the stroma, so it is coupled with the synthesis of 1 ATP molecule. So that is regarding according to the chemiosmotic hypothesis. So how that is possible will seem. So here the main thing is, so when this hydrogen ion has to move to the stroma, it has to pass through the thylakoid membrane, right? So but here what's the problem is, so in case of thylakoid membrane, it intakes the hydrogen ion, but it is impermeable for the outflux of the hydrogen ion. It won't allow the hydrogen ion to come out of it. Okay, so the membrane become impermeable. But in thylakoid membrane, there are certain regions called as ATPase enzyme complex or F0F1 complex, or it is called CF0C1 complex or ATPase enzyme complex. So which will be permeable for this hydrogen ions. So if this hydrogen ions has to move from the lumen of thylakoid to the stroma, always they pass through this ATP synthase enzyme complex. And as two hydrogen ions pass through this ATPase enzyme complex, so the energy generated, that is called as proton motive force. So proton motive force means, so it is the energy or force developed that is due to the movement of the protons across the ATPase enzyme complex. Okay, so that is called as proton motive force. So here, due to movement of the protons here, proton motive force will be generated, and proton motive force generated for movement of every two hydrogen ions, that will be sufficient enough to add a molecule of phosphate to the ADP in order to form the ATP. So that is how movement of two hydrogen ions or protons will be coupled with synthesis of one ATP molecule. So if that is the case, so we have 72 hydrogen ions. So for 2 hydrogen ions, we'll get 1 ATP. For 72 hydrogen ions, so we will be getting how much ATP? That will be 72 by 2. That will be almost up to 36 ATP will be formed. So the end product of light reaction will be 36 molecules of ATP, which is formed by capturing the light energy, as well as 12 molecules of NADPH2. And out of this 36 molecules of ATP, 18 ATP molecules as well as 12 NADPH2 molecules are used in the dark reaction for the synthesis of the carbohydrates, or it is also called as biosynthetic phase. So that is regarding the concept of light reaction in case of photosynthesis, which is coupled with conversion of incident sunlight or the incident light energy into chemical energy. So in the next part, we'll discuss regarding the dark reaction in detail.